Bob, you can come up front, that's fine. You know, the last shall not be least. Doesn't the Bible say something about that? The last shall not be least. So it's a pleasure to introduce James Sherley. Testing? Testing? Is this on? I guess so. It's a pleasure to introduce James Sherley, who is an assistant professor at MIT. He, in terms of background, originally from Tennessee, undergraduate degree at Harvard, an MD, Ph.D. at Johns Hopkins University, a little bit of a postdoc, then had an early faculty position at Fox Chase Center, and 6 years ago joined MIT in what's called the Division of Biological Engineering, but for all practical purposes, this is a department. I think he's doing some very innovative things in the stem cell area. And James, it's a pleasure to have you down here to hear about what you're doing, but also to let you see a little bit about what's going on down here in the South. So, James Sherley, welcome to Atlanta, welcome to Georgia Tech, and we look forward to your talk.
Thank you, Bob. Good morning. There are more seats up front here. Bob may call you by name if you don't come up on your own. So I want to first thank Bob for the invitation to come, and this is my first visit to Georgia Tech, and so far it's been very exciting. I've met some neat people, and I'm looking forward to talking to some others later in the day. So I think we'll get started. My talk today is titled A Biological Engineered Approach to Enabling Adult Stem Cell-Based Biomedicine. And hopefully by the end of the next 50 minutes or so, it'll be clear what it is we're trying to do at MIT in my laboratory with respect to that title. I want to start by just sort of learning how to use the controller here. I think many of you are aware, given the nature of this talk, that stem cells have been in the news a lot in the last 5 years or so because of interest in applying them to problems in biomedicine and biotechnology. I just use this slide to sort of introduce the concept that there are different types of stem cells that are of interest. The embryonic stem cells, which are derived from early embryos, indicated here, early blastocysts. There are also what are called premortal germ cells, which have properties of like embryonic stem cells that are derived from the gonadal tissue in the developing fetus, which I won't really have much more to say about today. And a large part of my talk will be about the stem cells that are found in adult tissues, called adult stem cells, that are found in pretty much every tissue in an adult mammal's body. So in biomedicine, there are many therapeutic targets that it is thought that stem cells might have applications for in biomedicine. I just list some of those here. Bone marrow, there's a lot of examples for applications, therapeutic applications for cancer in terms of bone marrow transplant therapies, blood formation diseases, and also gene therapy, where the idea is that a hematopoietic stem cell would be a great vessel to carry genes that could be curative. Bone and cartilage, there's interest in these cells for their regeneration properties, for developing new types of grafts. A lot of interest in the pancreas for approaching diseases like diabetes. In the brain, there's interest for neurodegenerative diseases like Parkinson's. Liver, there are not enough liver for transplant, so liver cell replacement is of great interest. And also in the case of liver, in drug discovery, there's interest for the properties of liver for metabolism and for toxicological studies. And then finally on this list is skin, for skin grafts, and because the hair follicle is thought to harbor the stem cells for hair, also for hair replacement therapy as well. So I've highlighted 3 categories here in white, bone marrow, liver, and skin, because those are 3 types of stem cells that my laboratory currently works on toward these types of applications. And I've underlined liver because I'm going to primarily tell you about our studies in liver today. So before talking about how stem cell might enable biomedicine, I want to spend a little bit of time talking about tissues and what tissues look like in the human and other mammals. And one way to think about tissues is that they are modular cellular architectures that integrate cell kinetics, the production of cells with cell function to achieve macroscale body functions. And that's a pretty general statement that we can apply to most of our tissues. I'm going to go through some of this briefly now, and I'll come back and address some specific aspects later, but the basic idea is that we have tissues that sit on, where there are cells that sit on basement membranes. These cells have the ability to divide with what are called asymmetric cell kinetics, which is a property of adult stem cells. There's a lot of interest now in the environment that these cells live in, but the basic idea is that they divide, they make another cell like themselves, and then they make a different cell type. In most tissues, these other cell types undergo symmetric cell kinetics, so they divide and make more cells like themselves. Eventually, differentiation takes place. And finally, there's a terminal arrest. The cells don't divide anymore. And eventually, they cease function, and by a variety of processes, including death, they are lost from the tissue. So tissues are constantly turning over. It's an important message here in terms of thinking about how one can approach cell therapy. Now, I want to give an example of this. One of the best examples is the epithelial tissue, in this case, the small intestinal epithelium, where this general statement applies. This is a section through the intestines of a rodent. The small intestines, it shows the tissue structure here, which has these dark blue nuclei, which form what are called villi. And then there are these regions in between that are referred to as pits, where cells are produced. If we look in schematic at this structure, it has all the features that I showed you earlier in my diagram. Namely, in this bottom compartment, what's called a crypt, there are adult stem cells. Their identity is difficult to prove, but there are adult stem cells there. They live in a niche there. And these cells divide in this region with asymmetric cell kinetics to produce non-stem cells, which undergo symmetric kinetics. There's a lot of cell expansion here. And in this particular system, there's a pretty sharp demarcation between where cells start differentiating. They differentiate, and they undergo a terminal arrest, and they're all lost out here at the tip when they undergo death and apoptosis. Now, most epithelial tissues can be described in this way. If you look at them closely, they all have this sort of basic architecture. What isn't appreciated often is that many non-epithelial tissues have the same type of kinetic architecture, and a good example of that is bone marrow. So what I've outlined here is a classical non-epithelial tissue, the bone marrow. It has a very similar kinetics architecture. So there's a hematopoietic stem cell. It undergoes asymmetric cell kinetics in its niche to give rise to what are called early progenitor cells. These cells divide symmetrically to expand the number. Eventually, the products of these divisions undergo differentiation to give rise to lymphoid lineage cells and myeloid lineage cells. And this also ends at terminal arrest and death. So one has to sort of think about those features of tissues when you're anticipating or planning to do cell therapy for those tissues. Now, what I want to do now is to spend a little bit of time delineating similarities and differences between the two main types of stem cells that are currently being looked at for enabling biomedicine, the embryonic cells and the adult. So embryonic cells are derived from embryos, as I said earlier, while the adult cells are derived from adult tissues. I'll refine that statement later. In retrospect, now, these embryonic cells are easily identified and isolated. We know what they look like. We know where to find them. You can get them out. In the case of adult stem cells, there are no currently known unique markers for adult stem cells. There's no marker that will tell you uniquely that's an adult stem cell and another cell isn't. Embryonic cells has a lot of interest because they are pluripotent, which means they have the ability to give rise to in development all of the mature tissues in the body. While adult stem cells are limited in their differentiation capacity, in some tissues they're multipotent. They can give rise to several different cell types, like the 4 cell types that are in the small intestines. In some cases they're unipotent. They only make one thing. One example with that would be the lens stem cells that give rise to the lens. Embryonic stem cells, the technology is very robust now. Many laboratories find it relatively easy to grow them. And I will talk a lot today about adult stem cells because they're hard to grow. Now, the problems that I'm sure you're aware of with embryonic stem cells is that there are moral and ethical issues with the destruction of human embryos to derive these cells. And a more scientific technological problem is that so far, when you take an embryonic stem cell and put it back into a mature host, you get tumors, and that's another barrier for their use. Adult stem cells have some solutions to these problems. Number one, you can get informed consent when you derive these cells from individuals. And thus far, in studies that have been done with tissues that contain these cells, for instance, bone marrow, those transplantations are not associated with the formation of tumors. So I want to spend a few slides looking at what does cell therapy look like with these two different types of cells, and what are the problems and what are the features of the planned cell therapy with these cells. And it's also will give the motivation for why my laboratory is elected to primarily focus on adult stem cells. So this is a cell therapy scheme using embryonic stem cells. This is our schematic for a normal tissue turning over. The problem we are confronted with is a damaged tissue or diseased tissue that's missing some of those modules, that are missing units. And the goal with embryonic stem cell therapy is to take human embryos and derive from them embryonic stem cells, and I mentioned the moral and ethical issues associated with that. Another approach, which doesn't involve destroying natural human embryos, involves what are called therapeutic clones, where one creates an embryo using an enucleated egg and a mature nucleus from an adult cell or adult-like cell. Now, in addition to the moral and ethical issues there also have been shown, at least in mouse models, to be many epigenetic defects in those cells, which may compromise their use for therapy. But at any rate, the plan with these cells is to develop them in culture, to turn them into mature tissues. And then these developed cells will be used to restore this tissue. Now, some of the problems and barriers here is that, number one, there are other cell types in those cultures. There's a lot of work now trying to control embryonic stem cell development. And the second problem is that there are embryonic stem cells that are still there. And as I mentioned earlier, these can give rise to tumors in the treated tissue. Now, one problem that is overlooked is the issue of turnover, because these mature cells may repair this tissue on short term, but not for long term. The repair will be short-lived, mainly because in the absence of putting in adult stem cells, you don't have the cell that produces the mature cells that you like to have in a continuous fashion. So embryonic stem cell therapy faces what I refer to as the adult stem cell bottleneck for cell therapy. Out of these developed in vitro cultures, one needs to isolate, stabilize, and expand adult stem cells. By providing them, you can get a fully repaired tissue by putting in the cells that are responsible for making these architectural units, and you avoid tumors. The other features of cell therapy with adult stem cells are outlined here. So the basic plan here is, here's the normal tissue that has received some damage or is diseased. One can take remaining wild-type units from this tissue and use them to expand adult stem cells in culture to replace the missing units. That's autologous. One can consider doing it allogeneically by taking a source of these cells from another person. And in both cases, this can be done with informed consent, so the moral, ethical issues don't apply here. The problem is that one has to expand these cells, make large numbers of them. And that's the problem that we are working on, because currently it's difficult to do. It's one of the main limiting factors to studying the cells and also to using them for cell therapy. So we have identified in our group 5 major challenges in adult stem cell research. They are, as I mentioned, expansion in culture. There are no unique markers. And it's not surprising if you don't have these 2 things in place. There's not a lot known about their specific cellular functions. Any cell that's going to be grown in culture for long periods, for therapy, one has to worry about mutations occurring, variant cells arising, so it's an issue of mutation-free maintenance. And a big problem, which we haven't confronted yet because we don't have the cells to do the therapy, is that we have to put these cells back in their natural niche. So niche engraftment is going to be another hurdle that we face. I put an asterisk by those barriers, those challenges that also apply to human embryonic stem cell research. So what I want to turn to now is, how do we address these problems in adult stem cell biology for the purpose of enabling cell therapy? And I want to start by really focusing in on the specific properties of adult stem cells. Now, the first point is that although we refer to them as adult, they appear, in fact, in the fetus. Late in fetal development, at about the time you can start to sort of see organs form, there's evidence for adult stem cells, and then they remain resident in these cells after birth. As I mentioned earlier, these cells undergo some very unique cell kinetic properties. So the first point is that in tissues, you may not have gotten this from my diagrams because there's 1 stem cell for every 5 non-stem cells, but in fact, these cells are very rare in tissues. Those columns I showed are usually maybe 4,000 cells, so they tend to be in the less than 0.1% range for a given tissue. They're said to be primitive. They don't express the fully differentiated properties of the non-stem cells, and they're long-lived. One can find tissues and cells in those environments for long periods of time. Now, the essential properties of adult stem cells that really define them is this property of asymmetric self-renewal by asymmetric cell kinetics. And what I mean by asymmetric self-renewal is that when this cell divides, it makes another cell of the same type. That's renewal. And the daughter cell is different. That's the asymmetry. So the renewal, the self-renewal is asymmetric. And this underlies the foundation of asymmetric cell kinetics, which refers to not just the stem cell's behavior, but the entire tissue unit. So the progeny of this division, one of these cells, will have the ability to divide for the lifetime of the organism in which it resides, whereas the other one gives rise to a lineage which is short-lived. So these cells here will turn over. And that's the asymmetry in cell kinetics. Now, the cells exist, adult stem cells exist in at least 3 different cell kinetic states. I've referred to asymmetric self-renewal. There are some arguments that these cells can spend time quiescent, not dividing at all. But they clearly can also undergo what are called symmetric self-renewal, a division event that produces not one adult stem cell, but 2. And these occur during normal tissue growth. If you think about a child becoming an adult, the child gets larger, the colon gets longer, tissues expand. There's a requirement to make more tissue units. One needs to make symmetric stem cell divisions. The other place where this can be observed is in repair. When tissue is damaged, there's a need to make new tissue units, in which case it requires a symmetric division by stem cells. This process is highly regulated. That's why it's in these 2 red brackets, because in its full extension, this gives rise to tumors. And there is a very tight link between the cell kinetics of stem cells and tumor formation. So what are the barriers that have been proposed for growing these cells, expanding them in culture ex vivo? There are at least 5. I've highlighted the one that we focus on, asymmetric cell kinetics, because it's usually not on this list. The usual properties on the list are, people have proposed that there's a loss of survival factors so that the adult stem cells simply die in culture. There may be a loss of factors that induce them to cycle, to proliferate at all. You can certainly think that there's going to be loss of cell-cell and cell matrix interactions. There's no question these things are lost. The question is whether or not their lack is responsible for the failure to expand the cells. And finally, it's been argued that the cells simply differentiate. They turn into something else in culture. What I want to spend time is showing you today that we focus on asymmetric cell kinetics, and we can say that it's at least one of the problems, and it may be the main problem for expanding adult stem cells in culture. So it's an interesting paradox in the sense that the property which defines adult stem cells we propose is the barrier to their expansion in culture, and that is asymmetric cell kinetics. And I want to go through what the rationale, what's the reasoning behind that statement. So the question is, if you take cells out of an organism and you put them in culture and they have this behavior where they divide asymmetrically, why is that a problem to producing them in large numbers in cultures? And I think when you have the problem presented in this fashion, it's very clear. What happens with a cell like this, if it keeps doing what it did in vivo, in vitro, is this. You make lots of cells over time. In vivo, these cells were typically lost to wear and apoptosis, but in your cell culture, they're still there. And so you accumulate these cells over time, while the stem cell dividing asymmetrically doesn't increase in number. So there's a loss of stem cells by simple virtue of a kinetic endpoint, is the hypothesis, the proposal. Now this is compounded by what cell biologists do, because we sample our cultures to maintain them. We come in and we take a sample, If over time you sample where there's an adult stem cell, the proliferative capacity of the culture is maintained. Because these cells are all eventually going to undergo a terminal arrest as they do in culture. But eventually, when you grab a bunch of cells where there is no adult stem cell, that is the end of growth for that culture. Right? So what our goal has been to do with this proposal is to find a way to overcome that barrier. And what I want to do now is to switch to the approach that we've taken to doing that. And I just want to take a moment to give one definition that I use for biological engineering, since I've said in my title this is a biological engineering approach. And that is the idea that there's a new discipline that applies engineering principles to elucidating the nature of biological systems for the purpose of engineering them. And I hope you'll see that flavor in the work that we're doing today. So here's the overall programmatic approach that we've taken to developing methods for expanding adult stem cells in culture by overcoming that cell kinetic barrier. We've developed in vitro models for asymmetric cell kinetics. We've also developed mathematical models around our findings in those in vitro models. So these are adult stem cell models. We've investigated mechanisms in them, as I mentioned. What we've learned here, we've applied to the problem of isolating the cells, of expanding the cells. When we succeed, and we have in several cases, we then have new adult stem cell models that are ex vivo models. They're not cell lines, we call them cell strains because they come right out of the animal. We investigate mechanism again, and the information we learn here, we reapply to the problem of being better at expanding cells in culture. And of course, along with this application, there's also a need to validate that we have adult stem cells. I'll tell you how we're trying to do that today. Optimization and also evaluation of them. So our in vitro model started with a gene called P53, which I studied, which I still do study, but I studied more some time ago. We made cell lines which had asymmetric cell kinetics based on starting with murine cells that had no P53 protein expression. P53 is a very important cancer gene. It's known to regulate cell growth, and the loss of P53 is associated with tumor formation. So we restored normal wild-type P53 function on a wild-type promoter, and what we learned is that when P53 was off, the cells had symmetric kinetics, and when P53 was on, they divided with asymmetric cell kinetics, producing a non-dividing cell and a cycling cell every generation. So within this model, we were able to learn how asymmetric cell kinetics were controlled, as I'll tell you later. And it also put the first idea in our minds that this type of division could be responsible for what's called senescence in culture. It's well known if you take cells and freshly explant them and maintain cultures over time, eventually all mammalian cultures come to an arrest. And that arrest is associated with loss of P53 gene function, almost invariably. We had found that if we took cells of that type and restored P53, we observed these kinetics. And that led to our development of a mathematical model to ask, could we really account for what people observe in population doubling studies by modeling based on asymmetric cell kinetics? And I won't have time to go through this in detail, but these data points here are what people typically see for cultures that undergo senescence in culture. You keep track of what's called a cumulative population doubling time. It's just a running tally of the number of times a culture has doubled versus time as you passage the culture. Typically what happens is cultures grow actively at first, and then they undergo a process referred to senescence, and it stops growing. So we could show that by modeling based on the idea that there were asymmetric cell kinetics with generation times for these asymmetric events that we, based on measurements that we made in cultures, that we could in fact very nicely, this red line is our model, account for that data with this idea. And from there, we argued that what we needed to do was to find a way to shift the asymmetric cell kinetics to symmetric. That was the answer. We call this reversible suppression of asymmetric cell kinetics, or SACK, referred to it as SACK through my talk. So if you start with cells that were explanted and your adult stem cell has this property, what you need to do is to make it grow symmetrically, making two stem cells, hold it in that state, and you also need this to be reversible so that you can reacquire the cells that you're really interested in using for therapy or for study. How do we accomplish this? Well, in our in vitro model, we could do molecular and biochemical studies to define what are the rate-limiting requirements for asymmetric cell kinetics. This is a summary of several years of work here. What we learned is that when we restored wild-type P53 function to a cell, this biochemical pathway, which is responsible for making all the riboguanine nucleotides in cells, which are known growth regulatory agents, this enzyme, this rate-limiting enzyme for that pathway, inosine monophosphate dehydrogenase, is downregulated. And that downregulation leads to a shift from what you typically see in culture, exponential kinetics, to the asymmetric kinetics that we define in our cells when we restore wild-type p53 function. The way we got to this pathway was by learning that compounds, such as this compound here, xanthocene, added to cells that have p53 restored, would overcome the shift to asymmetric cell kinetics. So if you have p53 on, you add this compound, your cells will shift from asymmetric to exponential. And it's reversible. If you take the compound away, they shift back to asymmetric cell kinetics. So from these biochemical studies, we recognized that we had the tools that we needed to test the hypothesis that the reason that adult stem cells don't grow in culture is because of these kinetic processes. So our scheme is here for suppression of asymmetric cell kinetics. We take cells that have just been explanted from an animal. Normally, we would say that these adult stem cells would encounter a dilution barrier. We add a SACK agent. There's several that we use now. This is xanthocene, xanthine, hypoxanthine. They're all in purine metabolism. They all lead to expansion of guanine nucleotide pools in different ways. And by doing that, we can induce the cells to divide symmetrically. We can get a clonal expansion of cells. We've also learned along the way, though I won't have much more to say about this today, is that high cell density is another SACK agent. Once you get enough stem cells, if you just keep them at high density, that will suffice to keep them growing symmetrically. And then the way the theory goes, if you take the SACK agent away, you lose the symmetric suppression. You go back to this mode of growth. And now you can study what these cells produce. So our first attempt at this was in adult rat liver. We basically took rat liver and did a conventional perfusion to get what are called non-parenchymal cells that contain a lot of small and different size hepatocytes and also non-hepatic cells. We did limiting dilution cloning and clonal expansion either in the presence or absence of our SACK agent. So these SACK-derived lines have the agent present, and the conventional cell lines don't. And let me just say, you always get cell lines out if you do this without the agent. It's their properties that are different. And so what we learned, if we looked at the way these cell strains that we derived grew, they have some very interesting properties consistent with our hypothesis. So this is a colony formation efficiency assay where we basically plate cells out at very low density so you can see the progeny of individual cells at about 2 weeks later. So you're looking at colony formation. And if we looked at our conventional cells and looked at their colony formation efficiency, it was pretty much the same whether the SACK agent was present or absent. So this SACK suppression ratio is about 1. There's really no effect. But these lines that were derived with the SACK agent present, when that SACK agent was present in this assay, they grew as well as the conventional lines. But if you took the agent away, there was a reduction in colony formation. And it varied anywhere from 5 to 40-fold reduction. Now, this is what you get if you stain with crystal violet and you hold the plate up to the light and you count the big colonies that you can see. But it turns out if you take these plates and you look under the microscope, there are just as many colonies there. It's just that most of the colonies in the off state are about 10 cells in number. So that predicted that what was going on is that we had a type of asymmetric cell kinetics, that a cell is dividing, making 1 cell per day, while in this state here, both cells that are produced are dividing, so one gets an exponential expansion of about 2,000 cells in 10 days, as opposed to 10 cells in 10 days. And we developed an assay to look at that. We call it the cystopair analysis for cell kinetic symmetry. It goes something like this. So what you do is you plate out cells at single cell density again. You let the cells undergo a single division event. You just basically wait until most of them are doublets. And then you can pulse them with this analog for thymidine called bromodioxyuridine. Any cell going through S phase takes up the analog, incorporates it stably into its DNA, and we can come back and detect that later using an anti-BRDU antibody that's fluorescent. So we'll see a fluorescent cell if it takes up the BRDU, telling us that the cell's in S phase. So this is a way of scoring whether we have asymmetry or not, because we already knew that when cells were growing asymmetrically, the non-dividing cells produced in our models didn't enter S phase. So the expectation for symmetric kinetics is that you'll find paired sisters where both cells are positive because they're both cycling, they both go through S phase. And in the case of asymmetric kinetics, one of the cells is positive, that's the cycling cell, and the other cell would be negative, that would be the non-cycling daughter. And as shown here, that's what we observe. This is Lig1 is one of these conventional lines. We're looking at it in the absence of the SAC agent. These are DAPI stains to show where there are nuclei in cells. So you see that there are 2 nuclei, 2 nuclei, 2 nuclei. And then the BRDU stain, the fluorescent green, tells us whether or not they've gone through S phase. And they've both gone through in general. And that was primarily what you see in a culture like that. Now, if you take one of the SACK-derived lines and you do the same experiment in the presence of the SAC agent, you see a similar picture where there are pairs of nuclei that are both green because they both are going through S phase together. And about 76% of the divisions that we observed are of that type. Now, if you take the agent away under the SAC-free condition, where I told you in the colony formation studies we were making about 1 cell per day, now what you see is pairs of nuclei, but there's only one that's going through the cell cycle, consistent with our hypothesis. So this is the first evidence that you could, in fact, recover from a normal tissue cells with this property, with this asymmetric property. And what I want to tell you briefly before talking about some of the properties of those cells, and the cycling cells more specifically, is that these cultures, in fact, do give rise to differentiating cells, as adult stem cells should do. If we take our agents away, we get more differentiation occurring. I won't spend a lot of time with this except for one point I want to make. We went into this in collaboration with Linda Griffith's laboratory to find cells to populate a liver bioreactor. We were looking for hepatocytic adult stem cells. One of our lines fits that criteria. It has inducible cytochrome P450 enzymes. It will secrete albumin. It has many properties consistent with being a hepatocyte producer. The method caught another type of stem cell that we weren't necessarily looking for. These cells are cholangiocytic. They make biliary epithelium, both by marker analysis and morphologically. If you take these cultures and you grow them up, and we maintain them in fetal bovine serum, you put them back in their homologous rat serum, and not with any matrix gel or anything, just in the culture dish, they make these sort of trabeculated structures. And if you look at those by transmission electron microscopy, they're not well formed, but they're biliary epithelium. So this is a section through one of those here in the transmission electron microscope. The cells have polarity. On the lumen here, there's a lot of little dots there. Those are microvilli. The cells are short and squat like biliary epithelium. So the method catches any cell type that has the kinetics that fit, irrespective of what the tissue happens to be. Now, what I want to turn to in my remaining time is what is the nature of the adult stem cells that we propose are in the culture? One of the things you'll see if you read the stem cell literature is that people get confused and start focusing on what the cells produce and not the stem cell. And one of the things that we want to try to do is not only to identify these cells, to study them specifically, we also want to try to find ways of identifying them. This is a real problem. So now we have an adult stem cell culture. You ask me, well, which one's the adult stem cell, James? I can't tell you. And so what we've been working toward is a way in vitro of identifying these cells. And the first lead to this, what I'll share with you today, was just looking at growth again. So a postdoc in the laboratory was actually interested in doing better differentiation. We don't have authentic hepatocytes. She's trying to make the best hepatocytes we can. So she started adding TGF-beta because it's a well-known hepatocyte differentiating agent. And we learned the following. EGF is also added in these studies primarily as what we thought was, in the early days, was a survival factor. It turns out it's not important for most of what I'm going to tell you. If you look at growth of these cultures, and you take the growth factors away, most cells in culture will stop growing, and that's what we observe for these conventional lines, Lig1 and Lig2. When you go from 10% serum to 1% serum, there's a consistent drop in the percentage of growth. If you add TGF-beta, you can reduce that even further. These are two independently derived SAC lines, the cholangiocytic line and the hepatocytic line. And what you notice here is that you don't get that robust reduction when you drop the serum concentration, and adding TGF-beta clearly doesn't give you the reduction that you see in the conventional lines. Consistent with this failure to arrest, we look at a molecular marker called proliferating cell nuclear antigen. In the conventional lines, it almost becomes undetectable when they are under the most induced condition. But you can see that in the case of these SAC-derived lines, it's quite robust in expression. There are cycling cells present. We've looked for those cells in situ using an antibody against proliferating cell nuclear. Oh, I'm getting ahead of myself. The earliest indication that something special was happening here came from just looking at the morphology of the cultures under the induced condition. And what we found is that there's a persistence of morphologically immature cells in the culture, even though we're hitting them with TGF-beta in high amount. So the bottom here are our non-SACK lines growing in 1% serum, so that reduces their growth. And shown here, you can see that the cells don't fill up the vessel because they basically stop growing before they reach confluency. Once we add TGF-beta, we get a marked differentiation of those cells in terms of flattening. For a SACK-derived line, the cells keep growing, as I've told you already. They fill up the flask. When we add TGF-beta, what we notice is that we get a inhomogeneity in terms of the differentiation. So we see regions that look relatively primitive adjoin to regions that look relatively differentiated. Now, one of the things I want to tell you here is that we've done transplantation studies in nude mice to show that these cells are not tumorigenic, so we're not looking at a transformed phenotype. Now, what we've done is to go one step further and ask, well, where are the proliferating cells in the system by looking for proliferating cell nuclear antigen? What we find is that in the case of a non-SACK line, as I've shown you, this is only a small sample, but in general, the cells are PCNA negative. All right, they're not proliferating when they're under the most induced differentiating condition. This is a control leaving out our primary antibody, showing that we get some signal from the secondary antibody. The non-SACK lines do show some PCNA-specific signal, but it doesn't have the characteristic nuclear intensity. In contrast, if we look at the SACK-derived lines, both lines, and again, these are DAPI stains to show you where nuclei are, you find these cells that have bright nuclei indicative of cycling in the presence of inducing conditions. So that's consistent with what the molecular studies were telling us. So now we had evidence for a kinetic cycling compartment that continued to cycle even though they were under differentiating conditions. And one of the questions we wanted to ask is, well, what about differentiation? Are these cells which are continuing to cycle, are they also differentiating? And the way we've approached that is to look for indicators of the TGF-beta response, and there's some very early ones. This diagram here shows that when the TGF-beta receptor is stimulated by the binding of TGF-beta, there are molecules referred to as SMADs. There's a SMAD3 and a SMAD2. They complex with other SMAD proteins and they go to the nucleus. And that leads to a cascade of events associated with differentiation of the cells. So we looked for this nuclear localization of these SMAD molecules as an indicator of whether or not cells were responding to TGF-beta. And we found the converse picture. So here's the non-SACK lines stimulated with TGF-beta. We're now staining for this nuclear SMAD2 and 3. And essentially, all the cells that we detect, greater than 80% of the cells, show that nuclear localization in response to TGF-beta. They're all responding and differentiating. In contrast, when we look at SACK-derived lines, we see some examples of very clear response, and then there are other cells that are negative. And so one question we've had is, what's the relationship between the non-differentiating cells to the cells that continue to cycle? And I'll tell you, there's a student in the lab right now working on the double staining. We have not been successful yet. But we've been looking at the statistical counting, and it's very consistent with the idea that they are one and the same. If we look at the fraction of cells in these studies that are PCNA-positive, it's about 32%. If we look at the ones that are SMAD nuclear minus, it's about 37%. We've done one other thing to try to link together the kinetics with the differentiation. We did a sister-pair analysis. So remember I showed you earlier in the sister-pair analysis that one of the daughter cells cycles and the other one doesn't in the case of the SACK-derived lines. And that's shown here for looking at differentiation. So our basic setup here is to do the same type of daughter pair analysis, but now we score for nuclear SMAD. And we ask, what is the frequency of symmetric divisions where both the cells are negative? We call these transit cell divisions or non-stem cell divisions where you get a differentiation event, both cells are positive. And we're looking for a rare subpopulation where one cell's positive, one cell's negative that will be consistent with the kinetics that we've seen earlier. And we find these events, and we find them at roughly the same frequency. These are some examples here. So this is a DAPI stain showing two daughter nuclei that are sitting right next to one another. And if you now let the cells divide, challenge them for about 30 minutes with TGF-beta, and then stain for nuclear SMAD localization, what we observe is that one daughter expresses the nuclear SMAD, the other one doesn't. Again, one daughter expresses the nuclear SMAD, and the other one doesn't. Now, if you look at one of our non-SACK-derived lines, for the same types of images, we find them. They represent about 10% of the cells that we observe are of this type. Currently, we're looking at that carefully, but we think of that as our chance occurrence. There's some probability of this event happening depending on the timing at which cells undergo differentiation. So we think of this as our background because these cells don't have any of the other properties that I've mentioned before kinetically. Now, when I look at the SACK-derived lines, the number that we get here, again, is about 34%. It's a 3.5-fold increase, which is some level of statistical significance. But what's sort of striking about this is under the same conditions, if you ask me, well, how many cells divided asymmetrically in the Kinetics study? It's about 24% of them. And it accounts pretty much one-to-one for the cells that we see in this culture above that background that have this asymmetric feature. So this is where we are now. We have what we think is our hypothesis is that this cell here that continues to cycle, doesn't respond to TGF-beta, doesn't show the early event, is in fact the adult stem cell in the system. And we're working hard to sort of bring more closure to that idea. So we think we have potentially a new way to think about identifying adult stem cells, which is basically looking at a very specific property of them, as I started out with. And that is that they divide asymmetrically, and they have a feature called, we call it adifferentiation. We're not the first ones to use this term, but we're the first ones to use it in this context. It turns out they're bacteria that don't change, and they're called adifferentiating bacteria. But the basic idea is that an adult stem cell, when it divides, it doesn't change. And it doesn't matter whether their growth factor is there or not. It retains its ability to produce another non-stem cell. So what we're currently working on in vitro is to look for this as an indicator of stemness. And we're thinking about this certainly in terms of building on what we've learned with TGF-beta SMADs, because looking for a negative signal is not a great marker to have if you want to identify cells. So we're trying to turn this negative finding into a positive method for detection. We're also going to be looking at differentiation properties that are a little bit farther away from the molecular signaling. That may not be the best direction to go in, but we're going to try some of that as well. And what I won't have time to tell you about today is that we've been looking at chromosomes in cells that divide with adult stem cell kinetics. We have good evidence that the chromosomes actually differ in their age between the adult stem cell and the non-stem cell. So we're looking at chromatin as well. So what I've told you today is this is an overview of our approach to adult stem cell expansion. I want to give you an idea of how we sort of take this new information from the in vivo studies and loop it back around and use it in expansion. So we're starting to get an in vitro view of what may be going on in the liver, certainly what one can put into culture. There are SACK-dependent cells in the rat liver. The ones I've described are one line which we think is derivative of a cell in the liver that can give rise to hepatocytes. And I want to be careful here because I didn't go into the details. This line also gives rise to cells which have a marker called CK7, which means that they're not, if you're familiar with oval cells, it means they're not oval cells, not the cells that are thought to show up in injured liver. But the fact that they're CK7 positive means that they may have some propensity to make biliary epithelium. That's a biliary specific marker. Only a small fraction of the cells produced by ligate have that property. And it may reflect the distribution of those two cell types in the liver. So this cell type here, though I've drawn it primarily as making hepatocytes, also has the ability potentially to give rise to biliary epithelium. What we didn't expect to find, and it may or may not be resident in liver, is this line, which we call ligate 13. It only does one thing. It divides and gives rise to cells that have cholangiocyte properties and hardly any of our hepatocyte properties. These are SACK-dependent. We think they represent cells which are dividing with asymmetric self-renewal in the liver. What about these SACK-independent lines? I remember I told you at the very beginning you always get lines out that grow. Well, these are all either biliary or they're hepatic, hepatocyte-like. And their properties are one or the other. And they don't have the kinetic properties. They don't show high levels of the asymmetric differentiation. And what we think they are is that they represent these cells mature in the liver, and by mutation, they become stable lines in culture. It's what you get if you don't suppress the asymmetric cell kinetics. These lines will in fact predominate. We've started looking at them, and as you might expect, we haven't done everything yet, but several of these in fact have p53 mutations. One of the questions we're asking is there a distinction in having p53 mutations here because that suppression wasn't there to give normal cells an advantage? Now, where we're going with the information we've learned about these cells is that we've learned is that these guys right here differentiate. You add TGF-beta, they stop growing. These cells here don't. So that gives us an opportunity to solve a technical problem in doing expansion, because the way we did these studies was by limiting dilution cloning. Lots and lots and lots of 96-well plates waited for long, long periods of time. What we like to do is to grab a bunch of liver and throw it into a dish and grow out what we want. This gives us an opportunity to do that, because the basic idea is take away the growth factors, grow out the cells that grow with your SACK agent. The SACK agent prevents the selection for these guys right here. It doesn't prevent them from arising, but allows you to get these guys to grow out, and then we can study them to look for the properties that are consistent with being adult stem cells. And that's what we're doing now. I just want to tell you what kind of progress we've made with this so far in terms of different tissues. So we started out in rat. I've told you about the rat hepatocyte adult stem cells and the cholangiocytic ones. We have now moved the project to ask, will the SACK method work in a different species such as the mouse? So we've attacked mouse hair follicle stem cells. These little blobs here are differentiating cells on a monolayer, what we think are our adult stem cells, producing sebum, which is a sebocyte-type stem cell line. We don't know yet whether it has any other properties, but that's our first capture from mouse. We've also found lines from strains from skeletal muscle that have these same kinds of properties of under differentiating conditions. There are these differentiating patches versus none, and we're characterizing those. We have our earliest strains from mouse pancreas. We're asking the differentiation questions first, but these cells have the asymmetric kinetics that I've mentioned earlier. And our first capture from human liver is shown here. Doesn't look too good right here. I haven't updated the file, but these cells are now growing. They're cryopreservable. We can maintain them, and what we're doing now is asking what are they, because the liver is a busy place. There are a lot of different types of cells that are in there. We don't know yet whether this is actually hepatocytic in nature, but it's the first time we've had a SACK-dependent human strain of cells growing in the laboratory. We're rather excited about that. So just to review, this is our approach where we build models, both ones that are real and ones that are mathematical, to use to investigate these sort of basic ideas about adult stem cell biology. We apply that to these studies to expand them. We do the same thing with our ex vivo models. We study them. We've actually done a little bit of modeling there as well. From this knowledge, we apply it back to trying to improve and optimize expansion. Just want to say a couple of words about validation and evaluation. There is a general sense that the only way to validate an adult stem cell, or any stem cell for that matter, is to put it back into an animal. I think that's very helpful to do, but it doesn't necessarily validate. It's an evaluation. So the biggest problem is that most times when we put things back in mammals, we have trouble figuring out what they're doing when we put them back. So we need to marry those experiments to in vitro validation as well, thinking about what are the properties that we would expect for these cells in vitro as we put them back in vivo for the purpose of finding out what they can do. Not necessarily telling us what they are, but are they going to be useful? Will they work in cell therapy? And those are the goals that we have currently in the laboratory. And then finally, I just want to make sure I mention the contributors to this work. The early derivation of the first SACK lines was really led by Ben Lee, who's now back at the National Taiwan University. Ben was visiting scientist at the time. A master's student, Josh Merrick, postdoc Jim Tonstad, and an undergraduate, Nicole Hatch, did most of that early kinetic characterization of the lines of the strains that I described. The later work looking at the differentiation and adifferentiation properties of the cells in culture was done by a very talented postdoc, Gracie Crane, who is now back in the UK. Krisha Penchalingham is the cell culture expert in the laboratory who's currently doing the work in humans. And Jean-Francois Paré derived those mouse cells that I didn't have much time to tell you about. The early liver work was done in collaboration with Linda Griffith in the Biological Engineering Division. We've gotten a lot of good help around MIT with mathematical modeling, things that we're still learning to do and how to apply best in our research. And again, Ben continues to collaborate with us from the National University Hospital in Taiwan. So I'd like to thank you for your attention, and I'd be happy to entertain any questions or comments.
Open the question. Marie.
So I just want to clarify your hepatocyte type and your cholangioductal kind of lines have expression markers for other lineages, but you haven't been able to actually force them morphologically down more than one lineage. Is that correct?
So the biliary lines have expression markers primarily for one lineage. So the liver is funny because biliary cells are known to express things like alpha-fetoprotein and albumin, which sometimes people think are hepatocyte specific, but they're not. But once you sort of get into things that are clearly specific, one line is pretty much cholangiocytic, and it's got some neat properties. It makes albumin, but it doesn't secrete it. Okay?
And the other question is, have you looked at NUM in your asymmetric divisions at all, since that's becoming such a hot, evolutionarily conserved asymmetric?
Yeah, we haven't. There's a long list on a board in the lab, actually, that we haven't looked at. Because it's an interesting question. A lot of the work on proteins like NUM has been done during development. While here, there's a type of development taking place, but it's post-embryonic, and it would be of interest to see if these proteins play similar roles in these types of cells, but we haven't looked.
In muscle, for sure.
Yeah.
The purine metabolite that you use to drive the cells, the symmetric division, I assume that those are present in the in vivo situation. And do you use like supraphysiologic doses to drive that, or?
So the main purine in physiological purine is hypoxanthine. And we're at comparable doses to hyperxanthine in about a third of the people in the room. Okay? The other two compounds, and actually the ones that, in terms of hypoxanthine, is the least active of these compounds. The other one, xanthine, exists in small amounts in the body, and xanthocene hasn't actually been described. It's predicted to exist, but it's such low, so they're very low levels in tissues. So in that case, for those two that work best, we're at super physiological levels.
If I can ask a naive question. You talk a lot about P53. So often in biology, there are parallel signaling pathways. Do you think there may be another factor molecule that could also do the off-on effect that P53 does?
It's a great question, and we have been thinking about it, though I don't have a candidate. One of the issues is that some laboratories are looking at expansion using pathways like Wnt, for instance, and sonic hedgehog is another pathway that shows proliferation in the adult stem cell compartment, at least in invertebrates. So one of the questions is, are those regulating kinetics in the same way, and are they doing similar things? And we're just starting to sort of look at that now, but I don't know. It's a good point. Could be.
Todd?
That was really interesting. I'll speak loud. That was really interesting. I was curious with the assay, the sister pair analysis, and that's analyzing, I guess, growth kinetics of the cells, assuming there's different types of cultures. Is there any way to incorporate into that model the differentiation that's occurring at the same time as the growth kinetic? Do you know what I mean? It's kind of a second order that's occurring in the same, and is there any way to kind of decipher that at the same time?
Yeah, what you'd like to do is to do multiple immunofluorescence analysis where you score for both simultaneously. It turns out, here's a place for some commercial development. Antibody compatibility is a real problem when you're trying to do double staining, because all the antibodies have been made the same way. So the detecting agents, the secondaries detect both types of antibodies. So you have to sort of engineer your own antibody to do these studies, and that's what we're doing now. Yeah, you could just simply do multiple, like in flow cytometry, we do 5-color analysis. You need to do that sort of in situ here. And the problem is that right now, for a lot of these molecules, they're not good reagents for doing those things. So we're trying. We've got another way we can look at BRDU uptake, for instance, that doesn't require an antibody. It's a Hirsch-Quench procedure. We're trying to marry that to the detection of SMAD 2 and 3. It's an essential question. We've got to figure out a way to do that. And there are likely to be some new things learned there, because I say these things are sort of, quantitatively they're similar, but it's going to be the places where you have the lack of overlap that will be very interesting to know about those. And especially in terms of timing, when does one, given that the cycling and the failure to respond to differentiation are happening in the same cells, how are they related to one another? Do you need to get the asymmetric event first to accomplish that or not? I took a slide out. We talk in the lab about not just asymmetric adifferentiation, but what is the asymmetric moment of the cell? It gets back to NOM. Some of these proteins you can actually tell in the cell before it divides what the future is going to be by the localization. Those are very important things to be looking at, and that would help us by having some type of double staining analysis. Good question.
Robbie?
I was just curious about the difference between embryonic and adult stem cells from the perspective of, so embryonic stem cells divide symmetrically, or is there a difference in how they respond to TGF-beta versus how the adult stem cells respond?
There are some striking similarities. Embryonic stem cells, in fact, don't respond to TGF-beta. They are known to be resistant. The mechanism is, in fact, known as an absence of one of the TGF-beta receptor proteins. We've been looking at TGF-beta receptor in these models, and it turns out that the proteins aren't missing, but their ratios are altered in the stem cell containing strains in a way that would be consistent with the failure to respond. What we haven't done is to show whether that's localized. We haven't demonstrated that that's a localization to the cycling cell yet. So there's that distinction. Another thing I think is important to realize is that in a lot of the studies that have been done to look at differences in gene expression between adult and embryonic stem cells have been confounded by people treating all stem cells as equals. So if you're looking for the stemness gene and you create your microarray study so that you look at things that are common between embryonic stem cells and adult stem cells, you will throw away things that are specific to the two cell types. And I think that's happened. And we've been looking at that as well. And it turns out that the genes that we find that are associated with asymmetric cell kinetics discretely define the genes that other people have identified as being adult stem cell specific, not necessarily the symmetric ones that are associated with the embryonic cells. So I think that the way to think about these cells is that they have some common properties like all cells do. They have a special set of common properties that reflect stemness, but they also have some distinct properties that respect their different roles in development and in adult life.
Any other questions? If not, James, thank you again.
Thank you all.